All right, welcome back, everybody. We will go ahead and get started with our second session today and final session for the symposium. If you are logged on for this session but weren't on for the first one, just a reminder that we will be posting a link to a Google form at the end of the session here. So if you need to record your attendance, if you're an NC State student, you can do it through there. And then at the end of the session, we'll also be doing our prize drawing for the free book and the gift card. Yeah, so that is all you need to know before we get started here. But I would like to introduce our speaker. We're going to be having about an hour for him to do his presentation, and then we'll do some question and answer at the end like we've been for the previous sessions. So can science provide evidence for faith? So far in this symposium, we have discussed several ways in which the two schools of thought can be complementary, but is it possible for science to provide circumstantial evidence for faith? As science digs ever deeper into the world around us, what does the big picture say? Might there be intentionality in how things in the physical universe work together? Well, exploring this final topic with us today is Dr. Fuzz Rana. He is the Vice President of Research and Apologetics at Reasons to Believe. He earned a Bachelor of Science in Chemistry at West Virginia State University and a PhD in Chemistry with an emphasis in biochemistry at Ohio University. He pursued postdoctoral studies in the biophysics of cell membranes at the universities of Virginia and Georgia, and has co-authored numerous papers for peer-reviewed journals, in addition to holding 2 patents. So welcome to the symposium, Dr. Fuzz Rana.
Hello, everybody. How are you guys doing? Good morning, good afternoon, and maybe even good evening to some of you. I sure hope everybody is well and is safe and healthy and is in good spirits as we continue with this symposium. And I also want to say thank you so much to the organizers for inviting me to be part of the program today. It's really an honor and a privilege to be part of a really impressive lineup of speakers. Let me go ahead and share my screen here. And I wish I knew how to do this a little bit more elegantly, but let me go ahead and share the screen. And let's see here. Is everybody able to see the screen okay?
It looks great.
Good, good. Thanks. I've had some problems recently with Zoom sharing my screen. So anyway, just wanted to make sure. I've been tasked today with the responsibility of addressing the question, can science provide a basis for faith? And I'm going to address that question from the perspective of someone who is a Christian, and also from the perspective of someone who's a biochemist. And then in addition to that, I also am going to interweave some of my own personal experiences into the discussion today. And when we take on a question like this, can science provide a basis for faith? There's really a number of other questions that are kind of umbrellaed underneath this larger question. For example, what do we mean by faith? How do we understand the concept of God? And is there evidence for God? What does that evidence look like if indeed there is evidence for God? And has God revealed himself to us? Has God made himself known to us? And if so, how has he done that? So these are all questions that we have to engage as we think about this larger question, can science provide a basis for faith? And what I want to do, first of all, is just tell you the first thing that really comes to my mind when I think about the idea of God as a Christian. And the first thing that I think about is the idea that God is transcendent. And of course, this means that he is independent and outside of his creation, that he's separate and distinct from that which he has made. And this idea of God's transcendence is really very important when we start thinking about questions like, can science provide a basis for faith? Because if you think about this, if God is truly transcendent, then in many respects, you could argue that God is inherently hidden from those of us who are his creatures, who are confined to this universe. If God exists outside the universe, is independent of the universe, then one of his endemic characteristics is that he must be hidden from us as finite creatures. And so today, in one of the major philosophical challenges to Christian theism is the hiddenness of God problem. If God really exists and he wants us to know him, then why does he not make himself more evident? And in some respects, one one to this hiddenness problem is the idea that God is transcendent. He's not hidden from us because that this reflects some type of divine evil, but rather he is hidden from us because of his very nature as being transcendent. Now, of course, it's not just simply Christianity that views God as transcendent. God's transcendence is very much part of Judaism. It's also very much part of the Islamic concept of God. But what's interesting to me is how this idea of God's transcendence plays out in Islam. Now, I'm not a Muslim scholar. I'm not a scholar, an Islamic scholar. I'm a biochemist. That's my academic training. But I do know a little bit about Islam because I grew up in a home where my father was a devout Muslim. And I even explored Islam as a teenager. When I was about 16 years old, I became very interested in Islam, and I recited the Shahada, which is to declare that Allah is the one true God and that Muhammad is his one true prophet. And I began to read from the Quran, at least the English translations of the Quran, and I began to pray every day. I learned how to pray from my father. And after about a year or so, I kind of gave up on Islam. There's a number of reasons why that was the case. But I know a little bit about Islam because of that experience. And one idea that's very important in Islam is this idea of God's transcendence. When you think about Allah as a Muslim, you think about God's transcendence as being one of his central features. And the implications of this for a Muslim is that God is unknowable. He's inscrutable. He is completely hidden from us. And that all we can know about Allah, according to Muslims, is that which Allah wills. We can't really know Allah at all. And in fact, to even go one step further and say that somehow we might know God or know something about God is considered to be blasphemous in Islam. And the reason why it's considered to be blasphemous is because you are elevating yourself to the level of God to say that you can somehow know something about Allah, that you're diminishing God's greatness in doing so. But in Christianity, this idea of God's transcendence is balanced with another concept, and that is the idea of God's eminence. That while God is, again, transcendent in that he is outside of his creation, that he is independent and distinct from his creation, God's eminence is the idea that God is present and active within his creation as well. And this includes within nature itself. This includes activity within human history and in our individual lives as human beings. And so this idea of God's eminence, in effect, balances the notion of God's transcendence. And if God is active and present within the world that we live in, within his creation, then it means that it may actually be possible to know something about him by studying, for example, the record of nature, by contemplating the world around us. And this leads us to another important concept, and this is the idea of revelation, that Christians believe that God has made himself known to us, that this God who is transcendent, who is endemically unknowable because of his transcendent nature, actually has gone out of his way to reveal himself to us, to make himself known to us. And according to theologians, this revelation takes on two forms. One is referred to as general revelation, which is God's revelation that is generally available to all people at all times under all circumstances. And this revelation includes the record of nature. It includes God's revelation through human history. And even you could argue it includes human beings as a revelation of God in the sense that we bear God's image. Of course, we also recognize that God has revealed himself in specific ways to specific people at specific times, and that this revelation is captured in the words of the Old and the New Testament. But we also recognize that it's ultimately through the person of Christ that God has revealed himself to us. So when it comes to this idea of the hiddenness of God problem, of course, the rejoinder is that from a Christian worldview perspective, God has actually made himself known to us in a variety of ways, that God has not remained hidden, but he has revealed himself to us. And this is where this concept now segues into the larger question that we're addressing today: can science form the basis of faith? Because um, if God indeed has revealed himself to us through the record of nature, and science is the investigation of the world around us, then we would expect that science should actually discover evidence for God's fingerprints, for God's handiwork within the record of nature. And if so, that revelation, that insight should actually point us to the reality of God's existence. And when it comes to my journey to faith, God's revelation through the record of nature was very important in my conversion to Christianity. As I mentioned, when I was a high school student, I seriously explored Islam. And again, as I said, I gave up on Islam for a number of reasons. And the net effect of that was I settled into a position of agnosticism. I didn't know if God exists or not. And I honestly didn't care as a teenager. And as I went off to college, I again espoused the position of agnosticism. And what was important to me as a college student was doing everything I could to prepare myself to go to graduate school to pursue a PhD in biochemistry. And as part of that, I took courses in, of course, chemistry and in the life sciences. And through that coursework was exposed to the evolutionary paradigm, this idea that evolutionary mechanisms alone could account for the origin and the design and the history of life, for the diversity of life, for the biogeographical distribution of life forms, that is, everything in biology could be explained through mechanism alone. And at that time, I reasoned, as I know many people do, that if biology can be explained through evolutionary mechanism, through natural processes, then what role is there for a creator to play? And so that perspective actually deepened my commitment to agnosticism. Now, when I went to graduate school, however, things changed rather dramatically in terms of my worldview. You know, as a graduate student, I was in an environment where I was now being encouraged to think independently, that it didn't matter what the consensus was scientifically speaking, but what mattered was what the evidence said, and that I was learning to be courageous enough to pursue the evidence where it led, regardless of, again, what was the consensus, what was the mainstream thinking. And on top of that, I was, of course, learning more and more about biochemistry through my coursework, through reading the scientific literature, through conversations with my fellow students and faculty members, and I even was beginning to do my own research. And it was through that process I really began to appreciate, in a way that I never had before, just how remarkable biochemical systems are. There's an elegance, there's a sophistication to these systems. You might even say there's an ingenuity to the way that these systems operate, in the way that they are structured. And I began to wonder, how do we as scientists account for the origin of these types of systems? This, in effect, is the origin of life problem. And as I began to read through the explanations for how life could have emerged through evolutionary mechanisms, I wasn't interested now in somebody just saying chemical evolution did it. I wanted to know the details. And as I explored those details as a graduate student, I quickly came to the conviction that there is not an evolutionary explanation for the origin of life, that chemistry and physics didn't seem to have the wherewithal to produce the types of systems that we see inside the cell. And it was at that point I became convinced that there had to be a mind behind life itself, that there had to be a creator. And that led me to questions like, who is that creator? How do I relate to that creator, if I relate to that creator at all? And as I began to pursue answers to those questions, I eventually found those answers, in my view, in the person of Christ, that it was through the life, death, and resurrection of Jesus that I could actually enter into a relationship with this creator whose fingerprints I discovered inside the cell. That, to me, the Christian worldview made the most condition. And so I embraced Christianity as a result of that. But the point here is that it was through the elegant designs of biochemical systems that I saw God revealed in the record of nature. And that revelation drove me ultimately to encounter the person of Christ through the words of Scripture and through a divine inner experience of Christ at the point of my conversion. So the answer to the question, I think, based on my own personal experience, is that science can indeed form the basis for faith. But what I want to do in the time that I have today is actually move beyond this intuition of design that I had as a graduate student, and to try to actually construct a much more formal case for a creator. Where again, we're moving from this sense of design, this intuition of design, to a much more robust case for design, a much more robust case for a creator. And in doing so, actually formulate this case for a creator within a framework that is scientifically robust, that could actually be considered to be a legitimate scientific conclusion. And this is going to be kind of radical for some of you to think that we could actually make a scientific case for a creator. And I've given this question quite a bit of thought. How is it that we can, or how can we go about establishing a scientific case for a creator? And I realize that there are a number of disciplines within science that actually are predicated on the ability of science to detect the work of intelligent agency within nature. And perhaps the chief discipline towards this end would be archaeology. Archaeologists are able to examine a rock and determine if that rock was shaped through natural processes or whether or not it was shaped through the deliberate action of an intelligent agent, of a hominin, if you will. And it's instructive to look at how archaeologists arrive at that particular conclusion, that an artifact is actually the product of an intelligent agent. And so what I want to do as a case study is talk about a recent or an interesting find a number of years ago outside of San Diego, California. This was a mastodon, set of mastodon remains that was discovered in 1992 by Caltrans, who was doing some road work and in the process unearthed the remains of a mastodon. And when the archaeologists and paleontologists visited the site, they realized that these mastodon remains were actually deliberately processed by a human, that these remains looked as if they were manipulated by a human. And so this now became a very important site. Now, at the time, nobody was able to actually date these remains. The techniques available for dating simply wouldn't work on the site. And everybody just assumed that this site must be somewhere between 10 to 20,000 years old, because this is approximately the time window in which modern humans made their way into the Americas. But in 2017, a paper was published where a team of researchers were able to finally date the site, and to everybody's shock, the site dated at 130,000 years in age. This is almost 110,000 years earlier than modern humans had made their way into the Americas. And so the researchers speculated that perhaps there were other hominins in the Americas prior to modern humans, and argued that maybe these were Neanderthals that actually manipulated this site, not modern humans. Now, this is an incredibly controversial claim. And in order to substantiate this claim, the researchers did a very detailed analysis of the archaeological site and presented a very carefully structured case for, again, Neanderthals in North America. And it's this case that is, and how they went about establishing this case, that is really of interest to us. First of all, they demonstrated that the site displays artificiality, to use a term I've heard SETI researchers use. That is, it had the appearance of being manipulated by an intelligent agent. And this was in the arrangement of the bones and the cobble. that it was an unnatural arrangement of the mastodon remains, and that there were rocks next to certain bones. And as they examined the rocks in detail, as they examined the bones in detail, they noticed markings that looked as if these were markings that were the result of the rock being used to strike the femur of the mastodon in order to extract marrow from the mastodon femur. They also, in addition to that, demonstrated that there were no other natural process explanations that could account for the arrangement of the bones in the rock. And this even included scavenging activity by wild animals. And then last but not least, they went into the lab and they took the fresh femur of a cow, the fresh femur of an elephant, and they actually struck those femurs with rocks, showing that they could produce the same kind of striation patterns on the bones and on the rock that they saw in the archaeological site. And on this basis, they concluded that these remains were actually manipulated by a hominin, and given the date of the site, that it must have been a Neanderthal or some other hominin that is yet to be identified that was in the Americas prior to modern humans. Now, as controversial as this claim is, that to me, again, isn't the interesting thing about this study. The interesting thing about this study is that it gives us a roadmap for how we can establish a case for a creator in biochemistry using the same kind of methodology that an archaeologist would use. So what we can do to build a case for the creator is ask these three questions. Do biochemical systems display artificiality? Can we account for the origin of biochemical systems through chemical evolution? This is, in effect, the origin of life problem. And then what does it take to create biochemical systems in a laboratory setting? And what does that tell us about the origin of biochemistry? So let's take a look first at the first criterion, and this would be the artificiality criteria. So the question is, do biochemical systems display an artificiality? And I think the answer to that question is yes. That one of the things that I find absolutely remarkable as a biochemist is that the defining features of biochemical systems are identical to those features that we would recognize as the work of a human designer. In other words, as we've studied biochemical systems, we recognize that these systems have certain hallmark characteristics that essentially define and describe universally biochemical systems. And it turns out that these are identical to the types of features that we would see in systems or objects or devices produced by human designers. And so we could argue by analogy that if these certain features reflect the work of a human mind and we see these same features manifested as defining characteristics in biochemical systems, this should suggest that these systems too are the work of a mind, that these systems are designed. On top of that, there are a number of biochemical systems that in their totality bear an eerie resemblance to man-made devices and objects and systems. And so this is what we'll be focusing on here in the next couple of minutes, is just some examples of these systems. But before we do that, in a sense, what we are presenting here is a revised and a revitalized watchmaker type of approach to establishing artificiality or design in biochemical systems. And this idea of the watchmaker argument, of course, I think is familiar to many people. This is the thinking of William Paley, the British natural theologian, who in 1802 published a book called Natural Theology, where he presents the watchmaker argument. And in my experience, many people misunderstand Paley's argument because what Paley did is he said, look, we have two objects here, a rock and a watch. And these objects exemplify two ways in which systems can come into existence, either through natural process mechanisms in the case of a rock, or through the work of an intelligent agent in the case of a watch. And so Paley asked the question, well, what distinguishes these two systems? And he noted that a watch has certain properties that again reflect the work of a mind. And so then Paley argued that when we look at biological systems, these systems have much more in common with a watch than they clock. And therefore, if a watch requires a watchmaker, then it's reasonable to conclude that life requires a creator. And so this is very helpful, a very helpful way to think and to reason about biochemical systems when we're in the process of trying to establish whether or not these systems display artificiality. And so what I want to do is, again, focus on two systems that that I think exemplify the artificiality or the design features of biochemical systems. One of them is a massive protein complex that is shown in this, this slide. And this particular complex is called F1 F0 Atpase, or ATP synthase is another term for it. And on the left, we see a ribbon diagram. On the right, we see a space-filling model. And this complex is known as a molecular machine. There are many, many protein complexes inside the cell that are machine-like in terms of their architecture and their operation. And again, one of the most extreme examples would be ATP synthase. And this is perhaps arguably one of the most important protein complexes in biology, that this is a ubiquitous protein complex that plays a role in generating ATP, which is the molecule that the cell uses to power its operations. This particular protein complex is embedded in the inner membrane of mitochondria in eukaryotic cells, and it's embedded in the inner membrane of bacteria and archaeal cells. So it's a ubiquitous protein, again, that is in effect an energy transduction machine that converts electrical chemical energy into mechanical energy into chemical energy. And this is another cartoon of ATP synthase, where we can see that embedded in the membrane is this cylindrical type of structure built from a number of different protein subunits. And within that cylindrical structure are these channels that protons will move through. And as they move through the channel, they will essentially activate the motor, a motor that will cause a rotor extending from the surface of the motor to rotate. And as it rotates, there's a cam at a 90-degree angle that will interact with the protein subunits that make up what's called the turbine. And that cam interacting with the turbine subunits causes mechanical movement that drives the production of ATP. So this particular protein complex converts electrochemical energy into mechanical energy into chemical energy that is then used to power the cell's operation. This is an incredibly elegant protein machine that operates at near 100% efficiency. Now, it's one thing to see a picture, it's another thing to see this protein complex in action. And so this is a computer animation produced by a team of biochemists that essentially interpreted all of the work that has been done on ATP synthase, and then produced a computer animation that depicts the movement of the different subunits relative to one another during the catalytic cycle. And as we watch this video, I just want you to note again the machine-like properties of this particular protein motor. So here we go.
Drive a rotation of the turbine within ATP synthase, resulting in the synthesis of ATP. Like all generators, ATP synthase consists of two separate motors coupled together. Within the membrane, we have the F0 motor, named after the binding of antibiotic oligomycin. And at the top, we have the F1 motor from factor 1.
So the point here is that this is, again, eerie in terms of how this machine operates and its similarity to man-made machines. This is, again, an incredibly sophisticated rotary motor that, again, is an electrically powered or electrochemically powered rotary motor. It's also instructed to see how biochemists who are non-believers react to ATP synthase. And one would be Nick Lane, who is a biochemist at the University College of London. And in his book, The Vital Question, he writes about ATP synthase, and he says, the most impressive nanomachine of them all, ATP synthase, is indeed a rotary motor in which the flow of protons turns a crankshaft, which in turn rotates a catalytic head. The protein works like a hydroelectric turbine. This is barely poetic license, but a precise description. Yet it is hard to convey the astonishing complexity of this protein motor. This is precision nanoengineering of the highest order, a magical device, and the more we learn about it, the more marvelous it becomes. Some see it as proof for the existence of God. I don't, but it is undoubtedly a wondrous machine. Now, another set of protein machines that I want to talk about today are those that manipulate DNA during processes like DNA replication, transcription, and DNA repair. And as it turns out that these protein machines that manipulate DNA are literally collaboratively working as a computer system. And to appreciate this, we need to recognize that DNA is an information storage molecule, and the information in DNA is really twofold. It's digital information in terms of the discrete nucleotides that are used to form the nucleotide sequence of DNA, and there's also analog information in terms of the three-dimensional structure of DNA. We're going to just set that aside for the time being. But to understand why biochemists now would argue that, again, these processes that are taking place inside the cell, again, are mimicking a computer operation, we need to consider the work of the British mathematician Alan Turing. Alan Turing is considered to be the father of theoretical computer science. And in the 1930s, I believe, he developed this concept called a Turing machine, which is an abstract machine that essentially forms the fundamental basis for computer operations. And for Turing, this machine consisted of an input and output and a finite control. And what Turing again envisioned was that you had this finite control that was able to take digital input that was fed into the finite control and alter that digital input in a prescribed manner to produce an output. Now, the finite control is only capable of very limited alteration of the digital data being fed into it. But what Turing realized is that if you took the output of one Turing machine and used it as the input for another, and you repeated that over and over again, depending on the combination of finite controls that you used, you could actually perform very sophisticated and complex computations. And this, in effect, is what goes on during the process of something like DNA replication, where you could think about the proteins that manipulate DNA as being like the finite controls that are taking the digital data from DNA, modifying it, and then producing an output strand that then is operated in turn by other proteins in the sequence of interactions that collectively constitute DNA replication. That is, DNA replication is in effect a computer operation. And again, with the DNA being the input and the output, and the proteins being the finite control. Here's another video produced showing what happens during DNA replication. This again is an animation, but think about what you're seeing in terms of Turing machines as you watch this video. So what we're looking at in this video is, in effect, a combination of Turing machines interacting to produce, again, an output, which is DNA replication. The similarity between the computer operations and DNA replication, as an example of manipulations of the DNA molecule, is so stark that it actually has inspired a new area of nanotechnology called DNA computing. And this is a picture of Leonard Adleman, who's considered the father of DNA computing. He's a computer scientist at the University of Southern California. And Adleman and others that are now working in this area literally build computers from DNA and the proteins that manipulate DNA. They're housed in little tiny Eppendorf test tubes that are about this big, and they are more powerful than the most powerful silicon-based supercomputer system that we've been able to build, in part because of the capacity for massive parallel operation But the point here is that, that this is really driving home the point that what we're looking at inside the cell when DNA replicates or when DNA is transcribed are literally computer operations. This is what Adelman said in an interview. The most important thing about DNA computing is that it shows that DNA molecules can do what we normally think only computers can do. This implies that computer science and biology are closely related, that every living thing can be thought to be computing something, and that sometimes we can understand living things better by looking at them as computers. So in other words, when it comes to two example biochemical systems here, ATP synthase and DNA replication, what we see is very clear evidence that these systems have the appearance of design, that they display an artificiality, in that they are highly reminiscent of the types of things that we would build, the types of devices and systems that we would build as human designers. So now let's move on to the second criteria, which is essentially the question, can chemical evolutionary processes explain the origin of biochemical systems, which again is the origin of life problem. And of course, you just had an outstanding lecture by Dr. Jim Tour highlighting the fact that chemical evolution cannot, as it's currently conceived, account for the origin of life. But this is actually widely recognized by most people that work in the field and people, of course, that are outside the field as well. For example, Paul Davies in his book, The Fifth Miracle, which is about the origin of life, said, "When I set out to write this book, I was convinced that science was close to wrapping up the mystery of life's origin. Having spent a year or two researching the field, I am now of the opinion that there remains a huge gulf in our understanding. This gulf's not merely ignorance about certain technical details, it's a major conceptual lacuna." And again, this is driving home the point that we really don't fundamentally have any understanding of how life could originate through chemical evolution. Hence, we don't understand how biochemical systems could have emerged. I'm not going to spend a lot of time talking about the problems connected with the origin of life, because again, Dr. Tour gave an excellent lecture towards that end. But what I just want to simply do is point out that no matter how origin of life researchers try to account for the origin of life, whether it's through a replicator first approach, metabolism first approach, or membrane first approach, each of these approaches winds up in utter failure. It's not just simply that we don't know how life could have originated. When we honestly look at the data, we have to conclude that at this point in time, everything we've tried winds up in failure. For example, when it comes to replicator first scenarios, and we don't need to know the details, there's a litany of problems connected with replicator first scenarios that could be categorized in these three problems: the monomer problem, the homochiral problem, and the homopolymer problem. In the book Origins of Life that I co-author with Hugh Ross, as well as in my book Creating Life in the Lab, we delineate some of these problems. But suffice it to say that these problems are so severe that, again, origin of life researchers in honest moments will acknowledge this. I remember a number of years ago now, I was at an origin of life conference, ISL, which stands for the International Society for the Study of the Origin of Life. And at that conference, the late Leslie Orgel was given the responsibility of opening up the conference with a plenary lecture. Orgel, when he was alive, was considered to be among the preeminent origin of life researchers in the world. And he was the father of what's known as the Rna World Hypothesis. And so he was asked to give a status update on where the Rna World Hypothesis stood. And after going through problem after problem after problem, he concludes his lecture by saying, "I hope that there are no creationists in the audience, but it would be a miracle if a strand of RNA ever appeared on the primitive Earth." When it comes to metabolism first scenarios, again, this approach is, again, replete with problems that I'm not going to detail. And again, in my book, Creating Life in the Lab, we discuss some of these problems. But Leslie Orgel was a critic, by the way, of metabolism first approaches. And in a critical review article that he wrote before he died, he said, "These scenarios require an appeal to magic, a series of remarkable coincidences, a near miracle." And a number of years ago now, I collaborated with a chemist by the name of Jackie Thomas, and she and I produced a critical review article where we demonstrated that membrane first scenarios, like replicator first and metabolism first scenarios, again, are replete with problems. So the bottom line is that we really don't know how life could have emerged through chemical evolution. So when it comes to the second criteria, we can say that we currently have no natural process explanation for biochemical systems. Okay, now let's move on to the third criteria. And this has to do, what does it take to create life in the lab? What does it take to go in the lab and to try to assemble chemical supersystems that would begin to assume the properties of a cell, the properties of life? And this work is really found in two distinct scientific disciplines, prebiotic chemistry and another area known as synthetic biology. So we're going to talk about each of these in turn. First, we'll take a look at prebiotic chemistry. And when it comes to prebiotic chemistry, of course, what's going on here is that researchers are trying to recapitulate the chemical evolutionary process that led from simple molecules to the very first protocells, or the very first life forms. And what they do is they go in the lab and they try to, again, develop an understanding of what kind of chemical and physical processes could in principle contribute to this, and how do these processes work, and then are these processes relevant to the conditions of the early Earth? And you might argue that on one hand, origin of life researchers have been successful in identifying, at least in principle, the processes that could have conceivably contributed to chemical evolution. But, and they've done a great job of detailing the mechanistic undergirding of these processes. Where they have miserably failed is translating what they've done in the lab to the conditions of the early Earth. And of course, the problem here is this, that in a laboratory, you have intelligent agents that are designing an experiment, and then they are executing that experiment under very carefully controlled conditions, where they're assembling an apparatus and they're adding the right solvent with the right pH and the right temperature. They're adding the right chemicals up in the right sequence at the right concentration. They are monitoring the reaction. They are maintaining stable conditions. They are stopping the reaction at the just right point in time. And through that, they can demonstrate, again, that certain chemical processes and physical processes could, again, contribute in principle to chemical evolution. But the problem here is that when we think about what happens under the conditions of the early Earth, it's a highly uncontrolled environment. And the fact of the matter is, origin of life researchers are now recognizing that when they work in the lab, they actually have incorporated themselves into the design of the experiment unwittingly. In other words, the researchers have become involved to the point that they have become part of the experimental design. And it's their involvement in the experimental design that actually contributes to the success of these reactions. And of course, under the conditions of the early Earth, there would not be organic chemists present to superintend the process, to supervise the process, to exert that type of control. And in fact, as you try to translate the chemistry from the lab to the early Earth, it's highly unlikely that that laboratory chemistry would ever be productive under the conditions of the early Earth. And this problem now is beginning to be recognized by origin of life researchers. Clemens Reichert in a review article says this: it's not easy to see what replaced the flask, pipettes, and stir bars of a chemistry lab during prebiotic evolution, let alone the hands of the chemists who performed the manipulations. And yes, most of us are not comfortable with the idea of divine intervention. And let me just illustrate this by talking about the RNA world. And I'm just, this of course is, in the minds of many people, the leading explanation for chemical evolution. And the idea here is that the very first biochemistry was RNA based, and that eventually this RNA world evolved to give rise to the DNA protein world of contemporary biochemistry. And there's a number of different that people cite in favor of the RNA world. One of them are studies that were initially done in the 1990s and continued to this very day, in which researchers can go in a lab and they can use clay as a catalyst in solution to generate RNA polymers from RNA building blocks. And the idea here is that on the early Earth, if clays were present, and they most likely were, that because of the interstitial layers of clay, you can actually get these locations where you could build up the concentration of RNA building blocks to very high levels, and then the clay could serve as a catalyst driving the formation of the polymer. And so the thought is that this is a way to explain how RNA polymers could have emerged on the early Earth under aqueous conditions. The problem is that when you examine the studies, you see that this work is done under highly pristine conditions, where the researchers excluded from the experimental design materials that would have been present on the early Earth that would either inhibit the growth of RNA chains or would cause the RNA chains to undergo breakdown once they formed. In addition to that, in these experiments, the researchers stop the RNA polymerization process before the RNA molecules become over about 50 subunits in length, because above that length, the RNA becomes irreversibly absorbed onto the clay. And if that would have happened under the conditions of the early Earth, those RNA molecules would be unavailable for subsequent steps in chemical evolution. And then last, in order to get these experiments to work, you have to buy the clay from a special supplier. The clay has to be treated in a special way so that it can function as a catalyst. And you have to use chemically activated nucleotides that would never have existed under the conditions of the early Earth in order for the reaction to proceed. In other words, RNA can form from RNA building blocks in the presence of clay under very precise set of conditions that have been jimmy-rigged at the get-go by the researchers themselves. That is, the researchers contributed to the success of this experiment. Paul Davies said this: as far as biochemists can see, it is a long and difficult road to produce efficient RNA replicators from scratch. This conclusion has to be that without a trained organic chemist on hand to supervise, nature would be struggling to make RNA from a dilute soup under any plausible prebiotic conditions. Simon Conway Morris goes one step further, and that's this, when he says that many of the experiments designed to explain one or other step in the origin of life are either of tenuous relevance to any believable prebiotic setting or involve an experimental rig in which the hand of the researcher becomes, for all intents and purposes, the hand of God. And so, in other words, it's not just simply that we don't know how chemical evolution could have produced the very first cells. It's that when we actually think about what is being done in these prebiotic experiments, we actually are providing demonstrable empirical evidence that intelligent agency is a critical component in the conversion of simple molecules into chemical supersystems that could assume the properties of life. And we see the same type of conclusion when we look at work in synthetic biology. Synthetic biology is the area of biology where the goal is to produce artificial non-natural life forms. And there's a number of reasons why researchers want to do this work. But as part of the work in synthetic biology is an approach called the bottom-up approach, where researchers are trying to build protocells in the lab, starting with simple chemical systems. And in many respects, what people are doing when they work in the bottom-up approach to synthetic biology is very similar to what people are doing when they work in prebiotic chemistry, is that both people are trying to create life in the laboratory, starting with simple chemical systems and building these chemical supersystems that again would assume the properties of life. And in fact, today, synthetic biology and origin of life research are intersecting each other in some very interesting ways, where many people that are working in synthetic biology are actually origin of life researchers, and many people that work in synthetic biology are providing some provocative insights into the origin of life question through their work. So these two areas, in a sense, are almost melding together at times in terms of what they are trying to do. But again, when we look at what does it take to try to create a chemical super system that assumes the properties of life, we see again intelligent agency is necessary. And I want to illustrate that by talking about a quintessential study towards this end, done by a team of Japanese workers, published in 2015. And these researchers in the laboratory created self-replicating protocells that could grow and replicate for multiple generations. They were even able to couple the replication of DNA in the interior of the protocells to the growth in the replication of the protocellular vesicles. And they even devised a way to actually provide nutrients to these protocells after the cell division process in order to sustain the replication for multiple generations. This work, in my mind, is really science at its very best. And this is a cartoon showing what these researchers did. And these researchers essentially created vesicles using special types of phospholipids that they extracted from living organisms. And they also incorporated into the vesicles a non-natural lipid that they designed and synthesized in the laboratory. This lipid was positively charged, and this is very important, as we'll see in a minute. And what they did then is they encapsulated within the vesicle essentially double-stranded DNA, a DNA polymerase, a DNA primer, and then some deoxyribonucleotide subunits. And then what they did is they heated the system very carefully, which caused the double helix to unravel, exposing single-stranded DNA that then was used as the template to synthesize daughter DNA molecules. But because the synthetic lipid they incorporated into the vesicle bears a positive charge and DNA is negatively charged, the DNA would now adhere to the surface, the interior surface of the vesicle wall. They then added another non-natural lipid that they designed and synthesized to the system. This lipid was incorporated into the membrane, underwent a chemical transformation, and that, as a result of that, caused the vesicles to grow to the point they became unstable and they naturally fissured apart to produce two daughter vesicles. They then had a different type of vesicle they developed called a conveyor vesicle that bears phospholipids with a negative charge, and that negative charge would interact with the positively charged lipids in the daughter vesicles to adhere to the surface. And then by adjusting the pH, they could get the vesicles to fuse, and voila, they regenerated the original vesicles that were then available to undergo another round and another round of cell division. So this is an incredible system that they produced. Again, science at its very best, but this work is all predicated on the fact that these are, highly knowledgeable biochemists that are relying on a couple of hundred years of accumulated scientific knowledge to develop a very elaborate strategy that they could then execute in the form of distinct protocols that were carried out by highly trained scientists working under carefully controlled conditions in a laboratory to produce this self-replicating system. In other words, this would not have occurred if it wasn't for the direct input and involvement of intelligent agents. And again, these researchers had to select the just right phospholipids with the just right set of properties. They had to design and synthesize two non-natural lipids to couple DNA replication with the growth and replication of the vesicle walls. They had to very carefully adjust the lipid composition of the vesicles that they formed. They had to very carefully control the laboratory conditions to even encapsulate the materials needed into the vesicle to form the original vesicles, the original protocells, I'm sorry. They had to very carefully heat the system and cool it back down to trigger DNA replication. They had to add the just right amount of lipid to destabilize these vesicles to cause them to fissure. And then they had to add the just right, they had to design the just right conveyor lipids that they could then get the fuse under the just right set of solution conditions to replenish the daughters with nutrients. Interesting to look at the language that these researchers use in the conclusion to their paper. To achieve this goal, we selected well-defined suitable lipids and macromolecules, including newly designed ones, and constructed a giant vesicle-based model protocell that links self-replication of information molecules with the self-reproduction of a compartment. In other words, they can't escape the fact that they have to use design language to describe what they have done. So, in other words, what we've done here is we basically said, look, the same three criteria that an archaeologist would use to establish the work of agency with respect to an artifact or an archaeological site can be used and applied to biochemical systems to show that these systems too must be the work of an intelligent agent. These systems display artificiality. We can't account for their origin through chemical evolution. And what we see is through work in prebiotic chemistry as well as in synthetic biology, that life requires agency in order to be generated, starting with simple molecular systems. And so I would argue that, that this criteria allows us to establish the fact that there does indeed to be an intelligent agent that undergirds life itself. And this then leads to the question, well, who is this creator? Who is this intelligent agent? And do I relate to that creator at all? And I would really encourage you to consider asking yourself these questions, and also consider the fact that the answer to these questions is ultimately found in the life, death, and resurrection of Christ. And it's remarkable to me that through the mediating work of Christ, that the creator that we see revealed to us through the record of nature is a creator that we can personally and intimately know. But I would argue that science can indeed form the basis for faith. All right, I'm going to go ahead and stop sharing my screen here and open up the floor, I guess, to questions.
All right, Dr. Rana, I have often heard scientists argue that those who appeal to a creator to explain life's design are guilty of a God of the gaps philosophy. Can you explain what God of the gaps means and how it relates to your model?
Yep. Well, the God of the gaps idea is this, that if we can't explain a particular feature in nature, then that we simply appeal to God as a way to account for that particular feature. And so we see it oftentimes utilized, or that approach utilized in Christian apologetics, where we say, look, we can't explain, let's say, the origin of life through chemical evolution, therefore God must have done it. So we're inserting God, at least that this is what the skeptics or the critics would claim, we're inserting God into places where we don't have current explanations. And the idea would be that when we develop those explanations, then God is squeezed out and natural process mechanisms replace God. And so I'm sympathetic to that criticism, but I would argue that just because we're critiquing an idea doesn't necessarily mean, and we're showing that there are shortcomings or limitations to the capability of natural processes to account for certain features in nature, it doesn't necessarily mean that inevitably science is going to actually explain how, how these systems could come to be and therefore squeeze out God. I think the origin of life is a place where, to me, the problems are intractable. These are just simply not, this is not a situation where give us a little bit more time and allow us a little bit more work and we'll figure out what's going on. I think the problems are fundamental and intractable. And so, you know, I don't think that the God of the gaps criticism necessarily has potency when it comes to the origin of life. But what I actually did in my presentation was not a God of the gaps approach whatsoever. I basically was using criteria that archaeologists would use to establish, again, the work of intelligent agency in nature. And so it was not a God of the gaps argument, but it was an argument based on a collection of criteria that if those criteria were satisfied, we could rightly conclude intelligent agency was at work.
Awesome. The next question is, what is a definition of chemical evolution?
Chemical evolution is equivalent to the origin of life or abiogenesis. And it's essentially the idea that chemicals will essentially, through natural process mechanisms, go from simple molecules to increasingly complex molecules that can then interact and further complexify to form aggregates that then would begin to assume the properties of life. So it's essentially the application of evolutionary principles to chemical systems with the idea that this could drive those systems from simplicity to complexity, and in the process again generate life forms through that process. We want to be careful not to conflate chemical evolution with biological evolution. These are two fundamentally distinct ideas.
If chemical evolution did happen in the past, why don't we see it now? How do people who believe in chemical evolution deal with this lack of observable chemical evolution?
Well, the argument that I've heard, which is not unreasonable to me, is that chemical evolution is only going to be possible under conditions in which there's no life that is present. Because once you have organisms present, those organisms are essentially, they're going to frustrate any kind of chemical evolutionary process, because they're going to be consuming the organics that would be produced that would be necessary to drive chemical evolution. So because many organisms are heterotrophic and they're going to consume the organic materials in the environment, the idea is that would essentially make chemical evolution impossible once life originates, or once heterotrophic life appears.
Why would it take so long for origin of life researchers to realize that they are part of the experiment apparatus and probably the active designer of the process?
Why would it take so long? I think it's, that's a good question. And I think, I'm not quite sure how to explain why it took origin of life researchers that long to recognize it. But let me point out that there were people that were part of the ID community that recognized this issue years ago. There's a classic book called The Mystery of Life's Origins, which I think you guys are giving away an updated edition of that. And I think that book was originally published, goodness, was it in the 1980s maybe? Walter Bradley, Charles Thaxton, Roger Olson, and they essentially pointed out the problem of unwarranted researcher involvement at that point. And I picked up on that and extended that idea in my book published in 2011 called Creating Life in the Lab. So it was, a decade after my book was published that I began to see origin of life researchers actually acknowledging that problem. And so I don't know why it took that long to do so, but it's encouraging to me to see that problem now being acknowledged.
Next question is, if DNA functions in the same way as a computer, is it like an electrical computer with chips or a mechanical computer with gears? And do you think that our familiarity with computers desensitizes us to how amazing that is?
Oh, that's a great question. It would be a mechanical computer, I think would be the best way to think about it. It's not an electrical computer. So I think the answer would be it's a mechanical computer, but I've never thought about it before until that question was posed. I actually think that it's because we're familiar with computers that we actually are now able to see the remarkable, we're able to see the fact that DNA metabolism is actually a computer operation. In my book, The Cell's Design, I talk about I make use of a revitalized watchmaker argument to make a case for a creator where, again, I point out that there's these remarkable similarities between biochemical systems and human designs, which allows us to make a watchmaker argument. But one of the things that I bring up in the book is that that if indeed the watchmaker argument is valid, that we would predict that as we discover and invent and develop new technologies, that we'll actually see evidence for those technologies already existing inside the cell. It's just that we can't recognize those technologies in the cell until we ourselves invent them and become familiar with them. So I think if we were unfamiliar with computer systems and how computer systems operate, we would not see the similarity between DNA replication and computer operations at all. So I think it's the fact that we've invented that technology that allows us now to see that technology. And the fact that we've gone through the exercise of inventing that technology helps us to recognize how much more impressive the technology is inside the cell than what we can produce as human designers.
All right. Was the product of the 2015 Japanese experiment that you described a live cell? If not, what was the product? How far is that product from the simplest cell? And could you describe what it means for a non-natural lipid?
Yeah. So what was produced was not a living cell. The way to describe it is, again, it's a chemical super system that is a protocell in that it consists of a vesicle that has material encapsulated within it. And what these researchers were able to do is produce these systems that began to assume some of the properties of a living system, which would be the capacity for replication, right? But again, that capacity was utterly dependent upon the manipulation in the direct manipulation of the researchers working with those systems. So it's a far cry from the even the simplest cell that we are aware of. The complexity of the simplest known cell is beyond comprehension. Actually, we recognize that the simplest cell must have a genome size of about 300 to 400 gene products. So we have an idea what those gene products must be, but we don't have a really good understanding of all the different interactions among the proteome of the simplest cell and how those interactions are critical for life. So what was produced in the lab is a far cry from what is the simplest life form. So by no means are these researchers close to creating a bona fide life form in the lab. But again, the work towards that end is highly instructive in terms of showing just how critically designed or how carefully designed these systems must be. Now, in terms of what is a non-natural lipid, it's essentially a lipid is a material that is hydrophobic. It's not water soluble. And that's a broad class of biomolecules. These are water insoluble components of the cell. And a non-natural lipid would be a lipid that doesn't exist in nature. And so these researchers literally had to design lipids that had particular or a specific set of chemical properties and physical properties in order to be able to couple, again, the DNA replication to the growth and replication of the vesicles themselves.
Can you comment on excitement around the latest Mars exploration and the hopes of finding life?
Yes, I can comment on that. Whether those comments are going to be more insightful or not is another thing. But, to me, I think, what NASA has achieved with the Perseverance rover is, absolutely incredible. I mean, it's science at its very best. It's a remarkable human achievement to think that we could build a craft and then transport that craft across our solar system and land it on another planet and then gather data. That's absolutely astounding. I'm not sure whether we're going to find evidence for life or not. If we do find evidence for past life on Mars, I don't think we can rule out that life actually was originated on Earth and was transported to Mars in the distant past. As I heard Dr. Tor say at the end of his Q&A, life is everywhere on Earth, and life has been everywhere on Earth for the last 3.8 billion years. And so if any time in the Earth's past there were impactors striking the Earth and it would sending ejecta into space beyond our Earth's orbit into interplanetary space, some of that ejecta is going to wind up on the surface of Mars. And if Mars is warm and wet at that time, that ejecta is going to be loaded with life forms that could essentially colonize the Martian planet. And so there's no reason to think that if we do discover life on Mars, that it wasn't actually Earth life that was transported to Mars, again, at some point in the past. Now, one other thing that I think is worth pointing out is when we think about the whole achievement of sending a spacecraft to Mars, that really highlights our exceptional nature as human beings. Because think about this, when human beings have been around, I believe, about 150,000 years on planet Earth. And when we first appear on Earth, we have an archaic primitive toolkit. And it's in about a period of about 150,000 years, we go from a primitive toolkit to being able to put a spacecraft on the surface of Mars, or a rover on the surface of Mars, where we can collect data about another planet. There's no other species in that period, in that duration of time, that has ever been able to, that has ever had the cognitive ability to do what we've done, and to be able to do that in the window of time that we've been able to accomplish it. By comparison, Neanderthals were on Earth longer than human beings were on Earth, and their technology remained static. It didn't change throughout the duration of their existence. And Neanderthals clearly were not able to do that, to do what humans have done. And there must be something distinct about humans that separates us or distinguishes us from Neanderthals. And I would argue that it's the image of God. It's the image of God which makes us exceptional as human beings, that allows us to put a rover on the surface of Mars. And so when we see these kind of human achievements, I think it's really important for us, I think, as Christians, to really revel and marvel in what the implications of these achievements are, because it's highlighting our unique and exceptional nature as human beings, and with the exceptionality being, I think, aligned with the image of God concept.
Awesome. Can you also speak on science of the gaps? Any gap in knowledge must eventually be filled by faith in science.
Really good question, and I think you see that played out when it comes to origin of life research, where there's, even though you see origin of life researchers acknowledging we have no explanation for how life originates, you see people like Paul Davies saying it's a major conceptual lacuna, people like Leslie Orgel saying the origin of life appears to be a miracle. In spite of all of these kinds of statements that are made, there's an absolute confidence that we're going to figure out how evolutionary processes generated life on Earth. There's an absolute confidence on the part of origin of life researchers towards that end. And that confidence isn't based on anything that we've done up to this point. It's just simply a confidence that's driven by a faith commitment to materialistic explanations and a particular worldview and a particular approach to science. And so that to me is an example of science of the gaps.
What do you think of the hypothesis that transposons, in regards to viral origin, are a mechanism of genetic change behind evolution? Would you say these transposons display artificiality or are a random natural event?
Yeah, well, I actually would take the view that uh, that that transposons are actually a part of the design uh, of the genomes of organisms, and that transposons, I would actually just take a different perspective. I don't think transposons are actually a viral origin, even though there's similarity between transposons and certain retroviral sequences. I see that as actually reflecting shared design as opposed to shared ancestry. And we're learning more and more that these so-called genetic elements that appear to be derived from viruses actually perform critical function in terms of gene regulation. And I am of the opinion that the activity of transposons and how it impacts the genome sculpture and genome structure is really in its infancy, or we're in our infancy in terms of understanding it. And I think as we develop a better understanding, we're going to see that it fits a design hypothesis much more readily than an evolutionary hypothesis.
That goes into our last question, which is, where do viruses fit into the discussion of the origin of life, if you want to consider viruses living? Are there any arguments about viruses being involved in the origin of life?
Yeah, the bottom line is nobody knows how viruses originate. And, I don't think of viruses as living organisms. I think of them as biochemical systems that can reproduce when they are functioning in a parasitic way, co-opting a host cell's reproductive machinery. So I don't see viruses as living. And uh, I don't think anybody really knows how viruses originated, let alone how they properly would fit into any kind of chemical evolutionary scenario. It's very difficult to envision viruses that would exist and would be able to replicate apart from, again, the existence of a host cell. So even from an evolutionary perspective, I think you have to argue that you must have had cellular life first before viruses would have emerged um, later as a later feature. But again, I don't think anybody really has a good explanation for how viruses could have originated.
All right. Well, I think that is all that we have time for here today. But thank you all so much for asking those intriguing questions. And Dr. Rana, thank you very, very much for giving your time to the symposium and for so thoughtfully and charitably going through that presentation and answering our questions here today. Thank you so much.
My pleasure.
Awesome. Well, everybody, thank you very much for participating in the symposium. We've really enjoyed exploring this topic here with you. Obviously, we're just scratching the surface, and there's lots more to dive into. We will be sending out links to everyone who has registered. The lectures are all going to be posted to YouTube, and we're also going to post them in podcast format as well, if you prefer to listen to them on the go. So be on the lookout for that. And then we're going to be sending out a document that has further resources if you want to dive in more we talked about so many different topics in the symposium, and we know that you can go in a lot of different directions diving deeper. So we will be sending out some recommended reading for you. So keep an eye out for that as well. But we hope that this symposium sparked your interest in the topic. And we also hope that you all have learned some things about science and faith. And I just kind of like to conclude with thinking about how Alistair McGrath suggested this way back in our first session about how science and faith might be able to work together, not only to help us improve our understanding of the world, but live meaningfully within it. So with that, I will close, but thank you all so much for your time for the symposium. We'd love to hear your feedback. But yeah, thank you very much and enjoy the rest of your day.